Good morning, and I hope all is going well with everyone. I'm sorry I'm not able to be there today, but I do want to thank the organizers for letting me have a chance to talk to you. Um, I think what I am going to tell you will have some interest with respect to gene therapy. So I'm going to talk about how factor VIII causes ER stress or protein misfolding in the ER, and that might lead to liver disease, including hepatocellular carcinoma. Here's a scanning micrograph of an electron, scanning electron micrographs that's colored, and you can see in the green are the mitochondria, and it's juxtaposed to these yellow lamellar stacks and vesicles. They're side by side. They communicate with each other. Protein misfolding in the ER causes ATP depletion, and that signals to mitochondria through calcium to increase oxidative phosphorylation to produce more ATP that is then shuttled back to the ER for folding. So there's quite a auto feedback balance to maintain ATP at optimal levels. Nothing to declare. So in terms of gene therapy hemophilia, there are two things that we're primarily interested, and that's to ask, why does AAV factor IX expression, why is it stable in the clinic over many years, whereas factor VIII expression declines after 1 year? And there are a number of issues to consider with respect to what I'm going to tell you. Is factor IX is expressed in hepatocytes, factor VIII is expressed in liver sinusoidal endothelial cells, and endothelials express von Willebrand factor, which stabilizes factor VIII in the circulation. And upon expression, factor VIII misfolds in the ER, but factor IX does not in a variety of cell types. And finally, factor VIII expression activates the unfolded protein response, or the UPR. So I'm going to summarize this briefly, but basically the UPR is composed of three arms. PERK is a protein kinase, IRE1 is a protein kinase, and ATF6 is a transcription factor. They are all held in an inactive state by binding to BIP, the binding immunoglobulin protein. And binding prevents their dimerization and activation for the kinases and prevents trafficking of ATF6 for the Golgi, where it's cleaved to release an active transcription factor fragment. So the BIP primarily regulates and shuts down the expression of these signaling transducers, shuts down their function. And in particular, when BIP is released, they signal. And one of the major signaling events results in EIF2 phosphorylation to attenuate protein synthesis, activation of ATF4 translation, expression of BIP and CHOP. BIP auto-regulates so that the more BIP you make, it will feed back and shut down the UPR. So this becomes evident as a number of years ago, we demonstrated that transient DNA transfection into the tail vein by hydrodynamic DNA results in activation of the UPR. And this just shows three different forms of factor VIII we use. One is the wild type factor VIII, and the other is a B domain deletion of the complete red B domain and 18 glycosylation sites. This is what's used in vectors because of its shorter size that could fit into AAV. And N6 is a molecule we made a number of years ago in which 226 amino acids from the B domain coming from here is inserted back into BDD. And we call it N6 because they're N6 N-linked glycans. And when you look at the expression of those after 24 hours of injection in the mice, you see wild type BD and BDD make expression of low levels of factor VIII, equal amounts of antigen and activity, and it accumulates in the liver, the antigen, whereas N6 is more efficiently secreted and you see little liver accumulation. And when you take those livers and analyze them, what you see is that the wild type and BDD activate BIP expression, EIF2 phosphorylation, as well as CHOP. And for N6, there's less activation of these pathways, but it's at a low level. And a number of years ago, Jody, you also published in it a while ago, demonstrates sorry about that, demonstrates that antioxidant treatment can improve the secretion of functional factor VIII from both wild type and BDD, but not for N6 because it's already well secreted. And it also reduces the amount of liver antigen. So antioxidants can reduce oxidative stress. They reduce UPR induction, and they also improve factor VIII secretion. So a number of studies have led to a publication last year. I'll just summarize that here, but factor VIII goes into the ER upon translation, and the A domain binds in particular to the protein chaperone BIP. BIP is a peptide-dependent ATPase, uses ATP to release factor VIII. And as a consequence, we found that BIP binds to this particular region in the A1 domain. We call it agron because it has a propensity of hydrophobic amino acids right around phenylalanine 309. And this region is necessary and sufficient for BIP binding and for activation of the UPR. If BIP does not bind here, actually what it does, it promotes factor VIII is aggregates, and it forms these aggregates that are insoluble. They have beta amyloid-like structures. They're not disulfide cross-linked. But if you make the cells happy, these will dissolve, and through a BIP-dependent process, it refolds. So the agron is both the primary BIP binding site as well as a hydrophobic site that stimulates aggregate formation. But what about aggregate formation in mice that are treated with these vectors? And this shows you the factor VIII thioflavin S stain, which stains beta amyloid structures, and the merge, the BDD transduced mice, you see factor VIII in the ER that's circulating around the hepatocyte nuclei. All those also light up with thioflavin S is shown in the merge, whereas N6 shows very little. So BDD does aggregate in mouse and hepatocytes. So in order to study the interaction with factor VIII with BIP, we introduce a flag tag into the C terminus of the endogenous BIP gene right upstream from the ER retention signal. And in this way, we don't alter the expression of BIP, which is important to study its interaction with other proteins. And we can IP with a flag antibody and ask what proteins are bound to BIP. And when we did this with injection of either vector BDD or N6, what we find is that we flag IP, we see the BIP flag coming down in these transduced mice, and very much you see that the BDD significantly comes down with BIP, whereas N6 does not. So it shows that this molecule can bind BIP, activate the UPR. We think these mice will be very useful because they're a model to measure misfolding of any protein, but in particular, factor VIII variants that you might want to study in vivo. So the next question came as to whether about human primary hepatocytes, can we study those? So we worked with Yippee the Zhang, who talked to you this morning, and he uses primary human hepatocytes, passages them in mice that have this mutation, so you can kill the mouse hepatocytes by NTCB intermittent cycling treatments, and that'll expand the human hepatocytes about 75-fold, where maybe 75% of the liver is human, and then they can isolate these hepatocytes that are mouse passage primary human hepatocytes, and then we get them to Yippee, where we infect them with lentivirus. And then we study the expression, and you can see with increasing amounts of lenti BDD, you get increasing amounts of factor VIII. That's good, we expected that. But you also see induction of BIP, induction of EIF2, phosphorylation, and also CHOP was not so shown well here, as well as other genes that we identified as UPR-induced genes downstream of factor VIII. So BDD expression does activate the UPR in human hepatocytes. So then the question comes around to what about human liver disease or murine liver disease? In the West, HCC, hepatocellular carcinoma, is driven by several factors which trigger steatohepatitis. And these are all listed here. Importantly, all of these induce ER stress in the liver, and they cause fatty liver. And initially, in response to all of these agents, you get fatty liver steatosis. They develop the inflamed state, the steatohepatitis, and eventually go on to develop hepatocellular carcinoma. So this progression of this disease initiates from ER stress at the very early stage, and then other factors intervene. So what is the role of ER stress in the liver spectral diseases? Transient expression, a number of years ago, we showed that Michael Karen that transient expression of MUP-UPA is a urine kinase driven by the major urinary promoter in a transgene developed by Jay Deegan many years ago. That's expressed in the ER and mouse hepatocytes. It activates the UPR, and eventually those expressing hepatocytes die after 6 weeks. They're overtaken by non-expressing hepatocytes that don't make UPA. At 10 weeks, we put them on a high-fat diet and measured their HCC development at 9 weeks. And they all develop HCC after 9 weeks, after that transient UPA expression. So the question is this unique to UPA, or does factor VIII have a similar problem? And so we went on and did that with measuring both. We measured N6 and BDD injection with vector, and this shows you the factor VIII plasma levels. It's greater than for BDD as we expected. And after 9 months of DNA delivery and 9 months of high-fat diet, you find that there are some tumors in N6 and more tumors in BDD as quantified here. The vector injected mice, we find no tumors, and N6, 7 out of 12 developed tumors, about half. And for BDD, all of the mice developed tumors. And this shows you some histology of those tumors, an adenoma from N6 transduced mice and an HCC from those mice. And you see in the adenoma, it shows the tumor here, and it's fatty, it's an exponent, that blow up of that section. And by a glutamine synthetase stain, which stains early HCC, but not adenomas, you don't see any adenoma stain. But in the HCC, you see dramatic staining of these sections that when blown up, you can see as indicative of an early HCC, typical of glutamine synthetase stain. So in conclusion, what we have is basically a summary here of which we saw the responses to all these factors upon transient DNA transfection in mice. The wild type in BDD are secreted at low efficiency. Wild type induces all of these responses, the UPR aggregation, inflammatory responses, ROS accumulation, oxidative stress, apoptosis, steatosis, and BHA promotes its secretion. And they, at least for the N6, we know that they develop HCC. And all that is much reduced for the N6 derivative. So in summary, BDD misfolds, aggregates, and activates the UPR greater than N6 in mouse hepatocytes, and it misfolds in human primary hepatocytes also. BDD binds BIP in vivo in murine hepatocytes, indicating it's misfolded, and that BIP binding probably activates the UPR. BDD induces more tumors in mice than N6. So can these attributes cause a decline in factor VIII expression in the human AAV clinical studies? And that question is for you to answer. These shows our collaborations. In particular, I want to acknowledge Zoji, Jing, and Judicorn, who recently have been working on factor VIII in the lab, which was originally studied years ago by Jody and Miao. We've been working with the German Research Cancer Center, Professor Heichenwalder, who's been studying the mechanism of hepatocellular carcinoma with us. Roland Herzog has been studying inflammatory responses, as well as providing AAV vectors for us. Yippee, I mentioned at Cornell, providing the human hepatocytes, and McGill, who's done all the histology and the pathology analysis, in particular Dr. Zhua Gao and his assistant Laudry, which have done the histology and characterization of the tumors. And thank you for your attention.